All right, so more about histamines here and allergies. So if you have allergies, you know about histamines and the sneeziness, watery eyes, runny nose, so itching, sneezing, fluid buildup, inflammation, and mucus, okay, coming out your nose. And Benadryl, which a lot of you may have used, act to block the effects of those histamines. Also sometimes with uh, allergic reaction, you get a rash, and that's also from the histamine. So this is really cool. This production, we need to learn about uh, monoclonal antibodies. Okay, very specific, purified antibodies that are made of these cell clones, and they're made to recognize one antigen only, and they use what are called hybridoma cells. So to make this, you need to inject whatever antigen you're trying to produce an antibody for into a mouse. So again, animal testing. We rely on animals for a lot of our medical advancement. So I know a lot of you are staunch animal rights advocates, and that's just a gray area that we have to, talk about and recognize that without a lot of the animals that we use, we wouldn't have a lot of treatments that we have. So we talked about challenge and response earlier. The mouse will make B cells that can make the desired antibody. So then we'll get a bunch of antibodies to fight an antigen that we're trying to fight off here. So we then remove the beta B cells from the mouse spleen and then combine them with our myelinoma cells, which are cancerous, which are so allow you to make a whole bunch of different hybridomas, and you want to make only the hybrid or only get the hybridomas that are make the targeted antibody, the antibody that the researcher wants. So targeted antibody, and then when they're isolated, once you have the correct ones, you have them clone each other. So just have them make more copies and copies, and they're put into fermentation containers. Where they secrete, basically keep them alive, feed them food, oxygen, so they produce all these monoclonal antibodies for us. And the diseases like malaria, they are used in HIV tests, and you probably heard of if an animal has rabies, you need to get a rabies shot immediately, and this is where the rabies shot comes from, using the monoclonal antibodies. They will then go in and hopefully stop that rabies infection. So here we have a little diagram showing how we make these monoclonal antibodies. So this is our antigen that is being injected into the poor mouse, and then we have antibodies being produced, and we see that right here. Look like little Y's. You see that in the diagram? That's going to be an antibody, and this is our tumor cell. And then we mix them together. Okay. Making our hybridoma cell. And again, we've got to make sure some of them will take up the antibody correctly or the antigen and then produce it. Some won't. So you gotta make sure only the ones that are making the antibodies are being produced or then put them into the fermenters, and they're gonna get cloned over and over again, make a lot of copies, and then we'll be able to harvest all those monoclonal antibodies from the fermentation tanks. And here is an image of showing these tanks. So all these cells are in here, living, reproducing. I'm not an expert on these, but nutrition is being delivered. The gas ratio, oxygen, CO2 is closely monitored. I imagine here at the bottom is where they might be removing fluid, where they can then purify the antibodies that they want. So smallpox, very important. We talk about this. The efforts to eradicate. Think about this: eradicating something from the world. 1950, the world got together and said, "Hey, we can do this. Let's go for it." 1967, everybody was on board, working full effort, and then in 10 years, 1977, the last known case was seen in Somalia. So that's pretty amazing. That in such a short amount of time, this disease that has really afflicted us for so long was eliminated. I mean, I think that's just amazing. And the reason, smallpox was a great test case to try to do this. For one, no animal vector, so it couldn't hide out in animals. If you got it eliminated from humans, then you were good. Okay, it wasn't gonna pop back up, some other time. Symptoms appear quickly. So once you get infected, you get sick, and that's important because then you can't spread it to other people. So symptoms appear quickly, so you can have treatment, and then what's called ring vaccination. So basically, people near infected person are then vaccinated. So the person gets treated, and then everybody around them, or they can't be treated if they're infected, but everybody around them is treated, so they can't spread it. And once you get the vaccination, it has long-lasting immunity. Vaccination is long-lasting. So it isn't like something like HIV or the flu that changes so fast. That's what makes the flu, HIV difficult to make a vaccine for, because it changes so fast. So still hasn't reappeared since 1977. So let's cross our fingers. It still exists in labs in Russia and in the United States. They keep vials of this stuff, at least in those two places we know for sure. But yeah, 1952, gone from America. 71 from South America. Europe very soon after the United States. Asia in 75, and it's interesting, and of course we said, in Africa was 77, but Australia doesn't show up here. I'm not sure why. Why they're not included. There's an asterisk, and I didn't see the explanation. But you see, you do see minor and major here, and basically major is more severe, and minor is, less severe. It's something like a 20, depending on your reaction to it, 24% death rate, and it's like a 1% death rate with the minor. So the major was more majorly destructive, deadly. And also, monoclonal antibodies are important for pregnancy test kits. So they test for, like we saw, or no, we're going to see in a video we're going to watch, HCG, human. That's what the embryo pumps out right when it attaches to the uterine cell wall. It gets the blood supply to the mom and releases this HCG to tell her body, please do not get rid of this endometrium lining. I need it. So if it exists in the urine, the HCG antibodies are attached to a dye as it flows down the test strip. So the urine goes through, the HCG picks up a dye, and then the little section that is for, if this lights up on the line, you're pregnant, you have immobilized antibodies. So there's antibodies there, and then it's waiting, and then that dye comes along with HCG, boom, it connects, it draws that line, and then that's you can see for an indicator for pregnancy, which then shows up as a line here. There we go. In the positive zone. And here you see the HCG, seen the pregnant. See how it attaches to this antibody that's waiting there? Or I'm sorry, it has this dye on there, and it's attached to a molecule that is designed to pick up the HCG. Then the urine continues down the test strip, and this is the immobilized antibody right here for HCG that you got through your cloning, and boom, the HCG sticks to it and then creates a line. And more urine, of course, continues and then attaches to another antibody that's just there to connect with that dye. And that's just there to make sure that, yes, urine did go all the way through. So if there was HCG, it would have picked up on this. It's like a positive control that everything worked. If you're not pregnant, there's no HCG to attach to the dye. And so this antibody for HCG isn't going to grab anything. So that dye is just going to continue to the end of the strip and then connect to that antibody that's just there to grab the dye and say, okay, test worked, but no pregnancy. And here's an example. And you can see here, positive, learn some German, schwanger. Okay, nicht schwanger is not pregnant in German. So the blue band appears when the HCG attaches to antibody. Okay, remember that HCG has dye attached to it, on it. Okay, so the HCG with the dye attaches that monoclonal antibody. It does not appear because if you're not pregnant, no HCG to connect to the antibody or attach. Let's see. We'll talk about monoclonal antibodies. So no HCG to connect to the HCG monoclonal antibody. D, why do we have that there? Just to show that the test worked. That the test worked as intended. So that urine flowed. Okay, the urine went through there and would have picked up the HCG if they were pregnant. Okay, so here we have blood types. So check this out. What we have here, these are all the samples of the different types. Here are the different blood types. Okay, so you can label this on yours. So this is your blood types. And up here, label this on paper. These are anti-A. These were all treated with anti-A antigens. So if there were any A antigens here, you'd see the clotting. This right here was anti-B. Anti-B. So any B antigens, it'll clot up. And this, you have A and B. Okay, so this, for example, this person is blood type A. So any A antigens are going to cause a reaction. You see the clotting here, basically, showing that there's A's present. Here, these will go after any B antigens. Nothing. No, so there's no B's here. Okay, and in this column, it has A and B. So definitely we have clotting, so we have either A or B. So because there was no clotting with anti-B, we know this person is blood type A. And we did get clotting when we had anti-A and anti-A and B. And you can see the pattern continue here. Look, wherever there's this anti-B antigen hitting, you have the clotting. Here, it doesn't have any A, so it's not going to react with the anti-A antigen. And anti-AB is going to react with everything because it doesn't. It's going to, the anti-A is going to react with B and the A and B. And this is a blood type O down here, and it's not going to react because it has no proteins, no A or B proteins. So it's not going to react with these serums. They're looking for these A, B, or AB proteins. So this is how you test your blood type. I did this in 7th grade to find out I was A. This is me, but can't do that anymore because of the issues with disease transmission potential anyway. Okay, epidemiology. If you look at that diagram or that it's actually 472 on in your book. There's a couple peaks. So, what could have caused it? You could have immigration from different areas. Immigration that could have brought in, say, from Pakistan or Bangladesh or other places. You could have had fewer vaccinations. Fewer vaccinations of the people in that area. You know, polio is known to spread. It spreads easier in the winter, so it could be the winter was worse. There could have been some kind of economic disruption. Oftentimes, if you see an increase, it means people weren't getting the health care they usually would get, and therefore didn't get protected from the polio or whatever disease or illness, and it can spread more. Unfortunately, you could also have some kind of conflict occurring, which makes people move around, less likely to get medical treatment. And why would they want to look at maps? And with maps, you can see patterns better that can explain, what's happening. And from those patterns, you can therefore design better vaccination programs. Vaccination programs. And why does the U.S. or anybody want to keep an eye on things happening? That's because we're all connected. We're all connected. So, whatever happens, can easily happen here. People can travel and, just basic ethics. Okay, we need to be concerned with people around us, all around the world, even if they're not, really close to us geographically. And here's some images I want to share with you. There's a lot of anti-vaccination pushes, and I'll talk more about this in class, because it's a real problem with people. It's becoming a political issue. So you see a lot of politicized ads and propaganda to try to dissuade parents from getting their kids vaccinated. Remember, this is why our, infant mortality rates have gone down so much, and why people live a lot longer, why we live more healthier lives because of these vaccinations. But people now don't see a lot of diseases, so they don't think about it as much. So obviously these are made to try to scare parents from getting kids vaccinated. But really what you should be looking at instead are, these are the effects. This is what happens. People in crutches, deformations in their limbs, from polio, as well as dying. These are lifelong. I know a lot of you, I've talked in class, about FDR, and that he was in braces in a wheelchair from his infection with polio in his youth. So again, here's this is little, we see, so this adult has polio, is in a modified iron lung here. And then this was a huge thing when Jonas Salk developed his vaccine, or developed the vaccine that eventually, in 1977, by 1977, brought an end to polio. Oh no, that was smallpox. I'm sorry. But eventually, that's one of the things they're focusing on, is trying to eliminate polio, just like smallpox. So here, typical, these are, this is a later infection. You can see these, if you look really closely, look, little donuts. They're like raised, and then they have a little dot in the middle. They're filled with cell debris. And typically, what this could lead to is diarrhea, pneumonia, and eventually, it depends on where it's at, but 20 to 40% death rate, like we talked before. Here's a little bit earlier infection. You don't have the raised bumps, but this is what you need to think about when you think about vaccination. And one thing that's been coming back too is whooping cough. And you'll see what happens is kids just can't breathe because they're coughing so much. And just think about being the parent of this child, or seeing this child in a hospital bed, barely being able to breathe. If you're lucky enough even to get to a hospital, a lot of people around the world aren't. And just sitting at home, your child not being able to breathe, that's a tough thing to have to face, and the potential death of that child. And then measles here, and this is also coming back. It was supposedly, there was in 2014, it was thought to be eradicated from North America. But guess what? 2015 through 17, it's come back, and they're worried about this year. I think there might have been some outbreaks already, and some people not getting vaccinated, people being convinced that it's a bad thing. But measles, people say, oh, it's no big deal, but it does lead to fever, which can lead to diarrhea and pneumonia, which are both can be deadly, especially to kids. And most of the people that die from measles are under 5, and I think there's around 70 2015, 70,000 deaths from measles worldwide, all preventable. Very inexpensive. It's just due to humans not doing the right thing. And this here you can see this poster. It's propaganda, but it's positive because it's showing this baby looking all cute and trying to get parents to vaccinate this baby.